Hello, everyone, and welcome to today's webinar, High Titer Adeno-associated Virus Production that is Cost-effective and Scalable. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Gibco by Thermo Fisher Scientific. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Chao Yan Liu, Senior Manager, Cell Biology at Thermo Fisher Scientific. Chao Yan, you may now begin your presentation.
Thank you, Michelle, very much for your kind introduction. I'm very glad to have this opportunity today to introduce our newly developed high-titer adeno-associated virus production system that is cost-effective and scalable. At Thermo Fisher, our mission is to enable our customers to make the world healthier, cleaner, and safer. We are the scientists, and our purpose is to enable our customers to fulfill their scientific purpose. That means to us, the success of our customers is our success, and we take the pride in our mission. Thermo Fisher Scientific does offer the end-to-end solutions that span the entire viral vector workflow, include the upstream process and the downstream process. We do offer superior products, services, and expertise to help our customers to develop breakthrough cell and gene therapies. Today, I'm going to focus on part of the upstream process, which highlighted here in red, include cell culture, plasmid transfection, and viral vector production. The Gibco brand AAV-MAX, the help-free AAV production system, is a scalable, high-titer with a simplified AAV production workflow. We could say is the cost-effective, based on the high titer. So this entire system includes the viral production cell line 2.0 and the viral production media, AAV-MAX transfection kit. This kit includes the transfection reagent, transfection booster, and enhancer. The system also includes the viral plex complexation buffer and the lysis buffer. This whole system, as the prototype product already available in the middle of February of this year, the whole RUO system will be launched in August this year, and with all validated performances. The Gibco brand the cell therapy system, the CTS AAV-MAX whole system, will be launched in next year, 2022. Why we call this system is help-free. So we use the triple transfection technology to co-transfect the 3' DNA and optimize the ratio to the 293 base producing cell line to produce AAV virus particles. And the mature AAV caspase protein will be produced. In next several slides, I will address each component in this whole system. In this system, as I mentioned earlier, we have this newly developed clonal 293F derived cells, along with amino original-free, chemically defined, protein-free, serum-free viral production media. This media is not only support the VPC 2.0, banking, recover, culture expansion, as well as support the transfection and the production. So the new AAV-MAX transfection reagent kit includes the 3 components, all chemical defined, amino original free. They work together. So in addition, the complexation buffer, this is the first time we have the chemical defined and amino original free. In addition, all these components, we also included polysorbate TWIN20 based lysis buffer in our system, just to provide the easy and convenient for downstream sample extraction. In this slide, I summarized some attributes of this new developed viral production cell line 2.0. This is a clonal cell line derived from HEC293F cells with the proof of clonality and clear lineage documentation. These cells, if you compare with the typical 293P cells, in absence of the SV40 large T antigen, really reduce the regulatory concerns. This new clonal cell line can grow in the viral production media and high density in suspension. The typical peak cell density around 12 to 14 million per mil in the shake flask, based on the initial seeding density. This clonal cell lines also produce much higher the viral titer compared to their parental cells. The doubling time of these cells is relatively similar to the parental cells, just slightly grow faster and a little bit shorter, the doubling time, averagely, below 24 hours. So the good feature of the cells is non-clumped phenotype and give you the robustness, the scalability and the great passage stability. Typically, we require a passage 30 and we tested beyond that. The documented cGMP banked cell line will be available together with our CTS, the whole system, in next year, 2022. In this slide on the right panel, you could see 6 images. These are the images we took during the single cell cloning process. On the top panel from the left to right, you can see the image before seeding with a clear background. That's the image we took before we seed the cells, but after we add the media. On the day 0, we seeded the cells and 0.3 to 0.5 per cell per well, and we catch the single cells. And the day after, just like 24 hours post the single cell cloning, we catch the 2 cells, the cells doubled. And we continue to take the image on the different day until the single cell expanded big enough, the colony, and ready to be transferred from 96 well plate to the 24 well plate and further down to the shake flask and for the screening. Here I show some feature with this viral production cells 2.0. On the left, on the top, I show the cells in the quick recovery post the thaw. These are 3 different cell bank 1, 2, and 3. We saw at the same time, and we monitored the viability and the VCD right post the thaw. Post the thaw viability is really high, around 97, 98%. The one thing I want to point out is the day 1. Post the thaw, the parental 293F cells usually show the viability drop between 5 to 10%. But this clonal cell line didn't show any viability drop post the thaw. This allowed our customer use these cells as quick as possible. They could start their production run in early as passage 3. Once they recovered the cells, expanded to the certain level, they can do the production immediately. The bottom on the left is average doubling time. I calculated this based on the routine cell culture, 3-day or 4-day cell culture, and the early log phase. And you could say cross the passage 8 to passage 48, the cells keep the similar doubling time. How about the stability of this cell line? We monitored not only the cell growth, we also tested the viral production and the different passage of these cells. In the middle panel on the top, you could see the growth curve analysis with different passage of cells from passage 12 up to passage 29. They show almost identical the growth pattern. On the bottom, we show the normal routine maintained cells. When you count the cells on the bicell or CDEX, you take the image, you could see the cells really distributed evenly, and the single cells is really not clumpy. There's a much better feature compared to 293T. And on the right of the picture is the 5 million cells, viability around 99, and you see the evenly non-clumpy feature. On the right panel, we tested all 5 serotypes for the passive stability based on the viral production. Here, I show you two serotypes as the representatives. As you can see, the cells can produce the viral vector and the early as passage 3, as late as passage 33. You do not really see the difference between the early passage cells and the later passage cell. They keep almost the same level of the performance. The second important component in the system is viral production media. This media is designed to support the VPC 2.0. This media is the AOF chemical defined and manufactured and the CGMP in our Gibco facility. Supports the VPC grow up to 12-14 milli per mil. But this media does require the supplementation with 4 millimolar GlutaMAX. The AGT powder format of this media will be available together with the whole CTS system in next year, 2020. In this slide on the right, I show you the growth curve with the VPC cells and the different scale in this viral production media, range from 125 ml shake flask to 2 liter. Lately, we also tested in 5 liter and the bioreactor because we run in different time and different assays, so I didn't incorporate that data in the same figure. But they do show the similar growth pattern. How about our transfection reagents? Is it suitable for the larger scale? So here I show you some attributes of this new transfection reagents. It is a lipid-based transfection reagent and needs to work together with the booster to achieve the high transfection efficiency and produce the high viral titer. We simplified the transfection protocol, made it adaptable and suitable to the larger scale transfection. In the middle panel, I show you pre-mixed AAV-MAX transfection reagent and booster, how stable this pre-mixed reagents. You can see less than 1 minute up to 2 days, and lately I tested even 2 months. They are stable and 4 degrees. So you do not really need to worry about this step as time. On the right panel, the light blue bar represents the complex holding time. Once you dilute your DNA, you premix your transfection reagent, a booster, you end the mix to dilute the DNA, how stable they are. So we tested more than 5 different serotypes. Some serotypes is stable up to 40 minutes, but majority serotypes show the best time being is between 20 to 30 minutes. Here I want to point out, if you use your plasma DNA, specially optimized, either too big or too small, you might want to consider to test the optimal complexation timing. How about the AAV-MAX Enhancer? How much the titer they could increase in this whole system? This enhancer is chemically defined, AOF, enhance the production of AAV particles in the VPC 2.0 cells. The addition of this enhancer is flexible. Here we show you can end the time of cell preparation for transfection, just give you a simplified the workflow. You do not need to come back and after transfection to do addition. But if you forgot, you still can do. And it's quite flexible. Within 4 hours, 5 hours is all can be done. So the effect of the enhancer on AAV titer showed in the middle panel. As you can see, in the presence of enhancer, the AAV titer almost double, more than double. And we further down to test the function titer, the transduced unit based on per mil. And you can see also the function titer also doubled in the presence of the AAV-MAX enhancer. Lastly, I want to talk about the lysis buffer. The lysis buffer is polysorbate, it's a 220 we usually call, and the base the buffer, and is very easy to use. We made a 10x concentrated. Basically, you just base on the volume and 10% to your express transfected culture, cool culture, and continue incubate on the 37 degree for additional 2 or 3 hours after 72 hours, on the day of harvest. Then you centrifuge and collect the supernatant as your lysate and move down to the downstream purification or downstream just like quantification process. In next several slides, I will show you the overall system performance as well as the comparison study with some competitors' transfection regions or competitors' culture media. Here, I briefly introduce the AAV-MAX transfection protocol. Here lists and the shake flask scale, but it is very easy to adapt to the large scale, such as the bioreactor. On the day before transfection, usually for the customer, they use 293 T cells, they need subcultured cells and before transfection. Because of the robustness of this cell line and the high performance of this viral production media, we could avoid that step. So you just maintain your cells as a routine culture, such as 0.6 for 3 days. Then on the day of transfection, you just dilute with additional fresh media, dilute the cells to 3 milli. Or you can culture 1.2 for 2 days. Then you dilute to 3 milli. Typically 1.2 or 0.6 for 2 or 3 days culture, the cells range about 4.5 to 5 milli. So you dilute to 3 milli. Even more, we optimize this protocol suitable for large scale. You could culture 1.5 milli per mil just for 1 day. And next day, the cells double because I mentioned doubling time just about 24 hours, a little bit less. So next day from 1.5 milli, they could reach about 3 milli, range from 2.9 to 3.1 or 3.2, all depends you do the transfection in the morning or afternoon. This way allow you to avoid to change the parameter if we run the bioreactor, keep the same volume for cell culture and for transfection. On the day of the transfection, the day 0, the step 2, you can add the 1% AAV-MAX enhancer to the 3 milli diluted cell culture. Then you could start to prepare the DNA and transfection region complex. As everybody knows, the diluted DNA in complexation buffer is quite stable. Typically, we recommend to use 10% of the complexation volume relatively to your transfection volume. Then in the step 4, you premix the transfection regions and the booster, as I talked before. Because this mix is quite stable, you can mix the time between 1 minute to 10 minutes. Then you add the premix the reagents and booster to your diluted DNA. Here, I want to point out one thing. Except the media, you would like to pre-warm before you dilute the cells. All other reagents, you could just grab from the refrigerator and directly to use. No pre-warm required. But the complexation reaction takes place in the room temperature, in the hood, for 20 to 25 minutes. Then you add the complex to the culture. In about 3 days, typically 70 hours plus minus 4 hours, you could perform the cell lysis or measure the AAV titer or just like directly move downstream the purification. How about the system performance? We did apply the system approach, try to integrate all the components to achieve the higher optimal performance. Here I show you 5 serotypes we used during the development work. Typically, the titer ranging from 1 × 11 or 1.5 × 11. So we use the GFP as a gene of interest during our development. based on the beta tester, our tester feedback, they use their optimized plasmid DNA constructs. They could get much higher the VG per mil. Some got 3 to 5 times 11, some got 7 to 8 times 11, and even one customer show over times 12. So this really is the plasmid DNA based. I want to point out the viability and the VCD and the harvest. The viability and the harvest typically range from 70% to 80% based on the serotypes. If the transfection is not efficient, or your constructs didn't work well, maybe the viability could be arranged about 90. That means the cells didn't produce enough, the viral particles. That's an indicator. So the VCD and the harvest ranging from 4.5 million to 5.5 million per mil, because we transfected on 3 million. The cells is barely double, less than double. How about the scalability? We call this the scalable system, and here I show you some data. We tested in the 24-well place, that's a little bit downscale, and tested in the bioreactor, and almost every size of shake flask. On the left panel, you can see the 24 deep well place. The volume is ranging 2.5 to 3 mils per well, and compared to the shake flask, 125 mils, the capacity with initial transfection volume of 30 mils. You can see the VG per mil is really compatible. In the shake flask, we tested 125 mils shake flask up to 5-liter shake flask. In 5-liter shake flask, the initial volume is 2 liter. So with addition, the 200 mils complexation buffer is about 2.2 by end of the run. So you can see across different size of the shake flask, we got comparable the VG per mil, the titer. Further down, we check the effectivity, the function titer in all these scales. In addition to the multiple well plate and the shake flask, we further tested the system in the bioreactor. It's a high performer finance bioreactor. The capacity is 3 liter. The final production volume is 2 liter. We compare with the typical 125 shake flask. So not only the titer is comparable, we also monitored the post-transfection growth profile and the bio profile, such as the ammonia, glucose, lactate, and the pH, and we see in the bioreactor, because well-controlled system, even more robust and more repeatable. How about the repeatability and the reproducibility of the AAV-MAX, the whole system? If you just check the VCD viability from run to run, it's very, very consistent. But when you run the qPCR, you do see sometimes between the time to time, run to run, and the operator to operator, you see the variation, something like 20–30%. Probably everybody believes the qPCR assay sometimes produce the variation, and not just like our cells, media, and transfection reagent, this performance. So that's why we try to further optimize in our qPCR assay to reduce the variation solely based on the assay. And you can see we tested 3 different serotypes, multiple times, and different operators. And basically, this system we could conclude is repeatable and reproducible. How to compare this whole system to other transfection reagents, other media in the market? So PEI-Pro and PEI-BAX are 2 widely used transfection reagents in the viral vector production industry. So in the left panel, you could see we use the same cells, VPC 2.0, same viral production media. We just replace the AAV-MAX transfection reagent kit with the PEI-Pro, PEI-MAX, or FACT-Pro. And you can see significantly the titer decrease when you swap the transfection reagent kit. So we would like to suggest is to use the whole system because we optimized each component to achieve the optimal performance. On the right panel, you can see the two popular culture media. One is Gibco FreeStyle 17. One is the Urban Balance CD Hec293 media in combination with PEI Pro. And you can see we would show more than 4, 5, 6-fold increase in titer if you use the complete AAV-MAX system. Finally, I would like to show some data about downstream purification. This purification for the AAV6 was done with the Thermo Fisher Go Pure AAV-X purification resin. Is all our Thermo Fisher reagents. As you can see here, is relatively not very large scale. Is a 1 liter shake flask production, around 250 to 300 mils production, loaded 200 mils. As you can see, is the shark elution peak, and with this 5 mils. And we calculate the recovered, the AAV6 particles, relatively is about 70%, the purified AAV6. If we run the purified AAV6-GFP on the SDS page, you could see the VP, the cascade protein, VP1, VP2, VP3 indicated in the elution fraction. We collaborated with our collaborator and the UCSD. They did transmission electronic microscope and show the image on the right and calculate the empty-full ratio of this purified AAV6. The full ratio is about 32%. The empty is about 68%. Right now, we really focus on the larger scale production, use this system, and the downstream purification with other cell types. And I think that's all I have for today. I'm really happy to answer any questions you might have. Thank you.
Well, thank you, Chao Yan, for your informative presentation. We will now start the Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. All right, let's go ahead and get started. So your first question is, how do titers compare to using PEI-based systems?
Oh, this is a really typical question because so many customers currently use the PEI. And as I show you the data in the slides, one is the transfection region comparison, one is the PEI in combination with competitive the media. So the titer is about 4 to 6-fold increase with our whole AAV-MAX system.
Okay, great. Thank you. Your next question is, how do the VPC 2.0 cells differ from the LV-MAX VPC cells? Okay.
LV-MAX VPC cells is viral production cell line, the first generation. That cells is not clonal cell line. It's just like the pool 293F cells adapted into the LV-MAX media. The VPC 2.0, the 6th generation, the viral production, the cell line 2.0 is true clonal cell lines, and screening based on the AAV production is the clonal cell line and the high titer cell line.
Thank you. Now, how much drop in titers do you see if no enhancer is used?
As I show you the data in the slides, with the addition of the AAV-MAX enhancer, you could double the titers, almost in all the cell types we tested.
Okay, great, thank you. Now your next question, when will the GMP version of AAV-MAX be available?
I as I'm aware, should be the second quarter next year, 2022.
Great. Now, are there differences between the RUO and CTS versions of the product?
Yes, the components in the system in the RUO or CTS should be the same. The CTS version, when launched, will be manufactured under CGMP and carries the benefits of the CTS brand. Means includes the additional testing with all the regulatory support.
Thank you, Chao Yan. Your next question, are there residual assays available for the components?
For the RUO, I would say right now is not available, but the residue assays for the transfection reagents and the enhancer will be available when the CTS version, the whole system launched in next second quarter of next year.
Great, thank you. We have one last question here. Are all the components animal origin free?
Yes, as I emphasized several times during my presentation, all the components in the AAV-MAX system are animal origin free and chemically defined.
Thank you, Chao Yan. Now, do you have any final comments for our audience before we go today?
I would like to say thank you to all the audience who take the time and pay attention to know more about this new system. And I would like to say one more time again, this system is really produce a high titer and we'll consider is really the cost-effective system. And the RUO, the whole system will be available shortly in the August of this year. Thank you very much.
Thank you, Chao Yan, for your time today. And we would also like to thank LabRoots and our sponsor, Gibco by Thermo Fisher Scientific, for underwriting today's educational webcast. Before we go, I would like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by our speaker via the contact information you provided at the time of registration. Now, this webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, goodbye.